we all have inside of our bone marrow an amazing cell type called a blood stem cell. And these cells are capable not only of dividing to produce more of themselves, but also differentiating to make the red blood cells that carry oxygen throughout our bodies, the platelets that allow us to clot when we have cuts, and the immune cells that protect us against infectious disease. And unfortunately, when we have errors or problems with this blood stem cell system, it can result in immune diseases and cancer. And so being able to understand how these cells are able to arise, renew themselves, and differentiate into these different immune cell types is really critical for being able to understand not just our healthy immune function, but also cancer and immune diseases. We used a technology called single-cell RNA sequencing, which allows us to take individual cells and measure the state of gene expression within each of those cells as this process unfolds over time. And in doing so, we could apply computational models to be able to predict which of the gene expression programs are actually telling blood stem cells which fates that they should adopt over time. So in other words, how does that stem cell know which type of immune cell that it's going to turn into? Some of the interesting findings from this paper is we're able to understand or learn about new genes that we didn't previously appreciate as being important for this process. So for example, we found one gene called YBX1 seems to be really important for allowing blood stem cells to become T cells, which are a key part of our adaptive immune system. One of the other findings that was somewhat surprising from this study is we found that the stem cells we're using in the lab are producing a cell type called mast cells, which in addition to being important for protecting us against pathogens, are also really important for allergic responses. Now, previously, people believed that mast cells came from a branch in this developmental process called myeloid progenitor. But what our data showed is actually that these mast cells seem to arise in a way that comes more closely with T cells, so a different branch of the blood development family tree than previously expected. So these are some of the findings from the study, but if you want to learn more, click the link below to access the full publication.